Okay, and so I realized actually when preparing this lecture, so that was good that I came here because I thought there were much more papers about that, and I realized there were not so many. So actually, that's a shorter part. So, and sadly, it's mainly our own work. So I don't want to advertise our own stuff, but there are very few people who are interested in this. So that's why I will just sadly have to show you our own papers. So the idea is that now we kind of were also interested if seed banks are affecting, the natural diversity, all the processes that I discussed. Do they also maybe affect the way selection is working, right? Because you delay many things happening, you have the storage of diversity which is dormant, so maybe it will also affect how selection works. Okay, so that was the idea behind this graph here, to understand what's happening to selection. Okay, and there are a few papers that have shown different ways of looking at that, and I will talk mainly about this part here. And it was an old paper actually from 1988, where people suggested this idea, and they look at a couple of data from some experimental work in Daphnia, and they conclude, yeah, selection must be slower because of the dormancy, but they did not explore that much further. So we thought maybe we want to get a bit more of a quantitative understanding of really what's going on there. Okay, so let's build a seed bank model with selection. So that's the little cartoon, how it works. So that's our paper. I will just write also a couple of the diffusion model that I use in the other papers without selection, just so that you see the difference. Okay, so the idea is that we use a kind of a forward in time diffusion framework. I think you had a couple of things about that, right, last week, a little bit. So the idea is yet to track in time the frequency of the alleles, right? So it's kind of the idea of the Wright-Fisher model, but now you use it in a diffusion framework where you assume population size is large enough, and you have this diffusion where the allele will diffuse in the population, right? And we want to see how long it takes until you can fix an allele or until it gets lost, for example. Okay, so how it works, and I think this equation already was hinted last week. So the idea is if we follow in time the frequency of an allele, let's say A, which I call little x, right, as a function of time. In the classic diffusion framework, you have a random effect, and this random effect is called a Brownian motion, right? So that's basically the random movement of the particle in the population, which is here the allele, right, so to say. And so this, in the case of the dormancy, what I showed you before, the model, you can write it exactly like this, because that's what the model is assuming. So I will write it here. So you assume that now we have this, I think you've seen last week, right? I think you mentioned it yesterday a little bit quickly. So this is basically, this is the diffusion term that you get from a diffusion process, and that's a random process. So that means you have your allele moving by genetic drift randomly in the population, right? In the case of the seed bank, it is affected by this rate beta, right? And if you remember, it was beta squared because here you have a square root, right? So in the diffusion terms, and so that beta is actually quite strong. And that's exactly what we recover if we use the model I showed you yesterday as a forward in time model. And so there are several papers I put on the Slack where you can show that there is the same process if you do it with a coalescent or with the diffusion. For the mathematicians there, this is called a dual process. So you can show it goes both ways, and that's quite interesting because you can match always all the models to show that they behave the same. So that's our model, and this is what I would call the Kai et al. Paper. That's the one I showed you yesterday. And if I now do the same process or the same diffusion for the model I just showed you before with these two populations, so this one, so that's what I will call the Blatt et al. Paper. Now we have two equations of diffusion because we need to track the allele in the two populations. So we need to track it in the active population and in the dormant population, right? So that's in the active population and that's in the dormant population. Here we have only one because we have only basically one active population only, right? If you remember, I didn't have two populations yesterday. Okay, and so what you can show is now that you have this rate C, as we mentioned before. I'm trying not to do mistakes because that's my favorite way of doing mistakes on the board. And then, and I'll write this one here. Okay, so what does that mean? Here you track the allele in the active population. Okay, so what can happen? You have a random process because you have the right feature happening in this active population. That's the same, so no magic here. But what happens is you see now that you can have individuals coming in and out, because some they go out when they go dormant, and some come in from the dormancy. And this is this term here. Okay? I'm just explaining basically the underlying assumptions here, right? And so if you track now the allele in the dormant population, what is going on? Well, you have no Brownian motion. There is no change because there is no right feature in the dormant population, right? So this term is not there. But you have, of course, coming in and out again due to the resuscitation or the dormancy. Okay? So that's what these two equations now, these couple equations, tell us what's happening in both compartments in the same time. Okay? Yep. Yeah, so I think it depends how you write it. So I must admit that in these papers I also got confused all the time. So I think it depends how you write, because sometimes you write if you would consider the individuals coming in or the individuals, where do they come from? And so I think that's how they write it in most of the papers. But I must admit that every time I'm always confused. So I'm happy to look at it again quietly. But it's one of the parts where I'm always confused whether this should be the other way around here.
Common term, like the one that you define.
Yep. Sorry, say again?
The addition term is a
yep.
fraction term, we should have different refactors. That's what you're saying.
Yeah, I think that's what, yeah.
I think it's okay.
I think we should write it carefully later, but every time I look at it, you get confused. So at some point, I just stopped looking at it. So, yeah, I think that's something that should write it carefully once to check it. But okay, so let's say that there are maybe some slight differences here, but otherwise, that's the general idea, right? So you check what's happening, and you can see that now we have only this random terms plus the migration between them, and so. What we did is we did a model which is a little bit different, because we tried to adapt the model of I showed you yesterday also as a forward in time. And so what we assume, which is a little bit different from this, but you will see at the end it comes to the same. We have a finite population of plants on above ground, right? So these are the tomatoes, which are just you see. And then we have now an infinite seed bank. So we assume that the plant do so many seeds that actually you never have a problem having seeds, right, coming in. Basically, for many plant species, that's also reasonable because they do a lot, and so usually you never have too much problem finding seeds. Okay, so the plants produce large amount of seeds. We have a population size n, which does not vary here, so it's also fixed. We have two alleles, and we want to track the number of plants which have this type, for example, big A, the allele big A, at any generation. Okay, and so what you can do is basically we define also the fraction of seeds which germinate. And so again, you can see that now we have this B function, as I defined yesterday, which is the germination rate of the seeds. So all the seeds automatically fall, right? You produce seeds. The rate of going dormant is 1. Basically, they all go in the seed bank. Okay? But it's just that they have now a different rate at germinating. Okay, so we kind of avoid this problem of defining the rate of dormancy. We just only define the rate of resuscitation, basically, here. And so we skip one of the parameters compared to the island model. And you can see that the total of seeds germinating is the sum of all the seeds that germinate with the allele A based on the function of germination of the allele A, big A, which is here the BAI as a function of the H. right? And of course, in that case, the seeds can stay dormant for a long time, right? So here we have a model which is also unbounded because the seed bank is infinite. And then, sorry. And then what we assume now is we put a little selection coefficient, which we call sigma, which is a weak selection coefficient for the allele big A. Okay, so we assume that now one of the allele has an advantage against the other. Then let's look what's happening. So we can calculate that now, basically we have a right fissure in the above ground population, right? So some plants will produce offspring. If they don't produce offspring, you always have some, I mean, they always come from the seed bank, right? So you draw from the seed bank. And you have a binomial sampling based on the alleles which are in the seed bank, right? So because we don't care about the plants above ground, every time we just say they are gone, and then we just sample from the seed bank again. And this is how you can write it. You can write this is a binomial sampling. The number of allele A is a binomial sampling of the seeds which are in the seed bank. And of course, you have this selection coefficient, and you can here maybe remember that it kind of looks vaguely like what you've seen last week with the selection, where you have the fitness here of the big A allele, and here the fitness, oh, there is a mistake here, that's the small A allele. Oops, that's a small A there. Okay. So basically the small a has a disadvantage. It has a 1 minus sigma coefficient, and the big A allele has a fitness of 1. Okay, and I will stop there for the demonstration. But by doing different rescaling of time and population size, we can scale it appropriately so that we can recover an equation of diffusion which exactly mirrors what we would expect under the seed bank model. So you can see, I hope that now we have this dxt, that's the frequency here of the allele big A in a population. This term is the one I wrote here, right? So I synchronize everything because the paper is completely differently written. So I synchronize the notation, that's exactly the same. However, there is here a term now, which is here the selection term. So that's the term which drives now the selection and tells me this allele, if it has an advantage, what will happen to it. And if you have a positive selection, you expect this allele will go to fixation, potentially, right? If it would have a negative selection, it would just probably get removed from the population. And you can see that now what is interesting is that here we have this beta factor, but in function, again, of the square term. So that means it need falling back on the beta square we had yesterday, intuition, from the neutral part of the model. But for the selection term, now we have a beta times sigma. And so we don't have a beta square here. So the beta is beta times sigma. That means the selection is indeed slower because beta is between 0 and 1. Right? So the selection is slower than what you would expect if you have no dormancy, right? So beta is 1, you fall back into the classic right fissure with diffusion, and a diffusion model with selection. In that case, you get sigma. That means you decrease the selection. However, what is kind of funny is that you decrease the selection, but not as much as how selection is affecting genetic drift. So I'm totally confused. So the seed bank, the dormancy is affecting selection, but not the same way as seed bank is affecting the rate of drift, because there you have a beta square indirectly. Okay. So I'll skip all the details. If you want the paper to look at it, I can send it also around. I must admit that Johannes Müller did the math, and I think he's also using a lot of sometimes heuristic and perturbation theory, so it's maybe not also how people in stochastic would demonstrate it. So and that's also one slightly different way of doing the derivations. Yep. Yep.
Is it by like the mean approximation? Because you can write the selection term.
Yeah, so I know. So in mathematics you have this drift term and the diffusion term, and so yeah. Go on. Sorry.
So is it because you're going from discrete to continuous time?
Yeah. So here we are only in continuous time. Yeah. And yeah. Exactly. Yes. Yeah. Okay. So I think in the neutral, I mean in the neutral model, this first part is not here. And you only consider in continuous time the diffusion process of this Brownian motion in the population, right? In a way. Okay, so I will stop now. I will not put more math afterwards, right? So that was the last equation, just so that we kind of have an intuition of what's going on. So When we look at this now, we were like, okay, that's kind of interesting because we were not expecting this. So what we were expecting is a beta square here, right? Because you would expect that now everything is delayed by beta square, right? So why would you delay selection by only beta, where you would delay the coalescent and all the neutral process by beta square? So that was a little bit counterintuitive to us. So what we did first is we did a couple of calculations in addition. We can calculate the time to fixation based on this diffusion. So that's the one without selection. So when sigma is 0, we fall back on what we have seen yesterday, right? The time to fixation, which is the same as the time to the most recent common ancestor, is basically scaled by this beta square. So everything is okay, right? That's the same model as I showed you yesterday for the neutral part. But now if you calculate the time of fixation of an allele on the selection when sigma is positive, you get this super ugly formula, right? So that's an exact formula. It just looks very ugly. The gamma is this Euler constant, and this is an exponential integral function. That's when you solve this diffusion process. This is how it happens, that you get some very complicated terms sometimes. The only thing that I would mention is that this looks nothing like a square, basically. And to convince you, this is how we drew it here. We have the time. And this is here the time of the fixation compared to when you have no, or the time, time to fixation. And so basically when you have B, here this tells you how big is the seed bank. So here you have no seed bank to very strong seed bank, or long dormancy, right? And we scale everything at 1, which is basically the Wright-Fisher. Okay, so we are at 1 here. This is the Wright-Fisher model where B is 1. We have no dormancy, and we scale everything by this number, right? It's this time to fixation here. And then what you can see is when sigma is 0, you have, this is the neutral model we had yesterday. You get indeed the square, this increase of the fixation time, because it is a square function, right? So that's okay. But now you can see that the one with selection here, when sigma is 2, I hope it's pretty clear that this is not increasing in square, right? Obviously, it's almost linear in B, kind of, not entirely, but yeah. You can see that, I mean, this is here would be 50, so it's not exactly linear in B, but it's for sure very different. Okay, so that's now I would say that this graph gives you an intuition that there is something going on here, that the selection works differently from the drift term, right? From a drift, neutral drift, okay? Okay, so would you have any biological intuition? Why is this? Because I think that's the most interesting at the end after the math, right? Is to think, why is this happening here? So we had an intuition for the neutral, right? You need 2 lineages to coalesce. They need to coalesce in the same time to find the ancestor. And I think one of you yesterday said, well, obviously it's beta times beta, right? But now here for selection, we have only beta. So why could this be? So one of the argument could be in the coalescent framework that basically selection will affect only one of the lineage. So you basically only affect the plants which are above ground producing many seeds at some point, right? From the more biological intuition, I think it's not very trivial what's going on there. I think, and I have to say that still to that day, I don't have really a perfect explanation. So that's why I was hoping someone would come up with one, which maybe is easier than mine. The idea is really that you only have selection above ground, right? Because the selection happens only when you produce the seeds, right? So there you produce the seeds. So if you're fitter than another plant, you produce more seeds, right? But it only happens when you're outside. Once you're in a seed bank, basically nothing happens to the seeds, basically, right? So there is no selection in the seed bank here about, well, the seeds can get eaten or whatever. And so the intuition would be that only selection happens only in one part of the life cycle, and actually the rest is frozen in the seed bank without anything happening to it. But I must admit that this is not the most intuitive explanation I've ever came with. So if you have some intuition or you want to discuss about it, I'm happy to maybe discuss about it again. But it is an interesting phenomenon. And you can also show that the side frequency spectrum, again, that's the same graph I showed you before. Without selection, that's the neutral side frequency spectrum. It follows exactly the Kingman when we rescale. That's what we did before. And here, what we show is the side frequency spectrum with selection. And you can see when you have no seed bank that's in black, that's the expected under the Kingman in the Wright Fisher model diffusion with weak selection. You can see that the side frequency spectrum is barely affected. But the same with the same coefficient, when we have now dormancy, you can see that the side frequency spectrum starts getting more U-shaped. So seed bank is increasing the effect of selection, so to say, so you get even stronger signal of selection than what you would expect under the model without seed bank. So that's a bit counterintuitive, right? Because the seed bank is slowing down everything, but the effect of selection seems to be stronger. I still don't understand everything, right? So still things that we are still trying to process. Okay, so what is a bit of an intuition was provided in this paper by Shoemaker and Landon, where they did some simulation, but they did very few simulations, so it doesn't entirely help, but then they use the same model with the two demes I showed you from Blatt. And what they show is basically that you have here the frequency of the allele, which is on the positive selection. Okay, so they go from 0 to 1, and that's the classic selective sweep that you have seen last week, right? So you have selection, you fix the allele. You can see that when the time in the seed bank, when you have a weak seed bank, the time is actually longer than what you would expect without seed bank, but it still reaches fixation very fast. Once the time of the seed bank is increasing, you see that you have 1,000 and 10,000. You see that now the frequency of the allele in the population has this kind of weird shape, because first it's increasing, but then at some point here you have a lower rate of increase until it reaches fixation, and it takes way longer, right? So you can see that the time now here is in log. So you can see that you have a huge difference in amount of time until fixation. So what is the intuition? I think that's a bit more intuitive here. What is going on? The seed bank is slowing down selection, so it takes longer because, of course, you have selection only in the active population again, right? So you have your bacteria, they compete, and this allele will reach fixation in the active population, but then you have all the dormant bacteria which still don't have this allele, right? And that means now the allele has to kind of, so to say, wait until all the dormant bacteria get awake again, so that they can be on the selection and then reproduce or not, so that the allele can fix in all the bacteria in the population. So in a way, you have a first phase where you fix in the active population, in all the active bacteria, but if there are many dormant, you need to wait until, so to say, all bacteria, including the dormant ones, are also fixing this allele. Right? So in a way, you have a two compartment, and that makes sense in this compartment model, because first you fix in the allele in the active population, and then you have to wait now until, so to say, the allele can fix in the dormant population. But it takes much longer because in this dormant population, you need to wait until the bacteria become active, right? And so it can take a long time. And that's why they find this two phase here in the time. And you can see the time to fixation is here with the average time in the seed bank is really increasing a lot when you take in the dormant compartment. The yellow is the active and dormant compartment, the average, and in the active compartment, you can see that this is always reasonably fast. The simulations were not very extensive, and we were not very happy with this kind of explanation, so that's why we then decided to do our own simulation, do our own model to test a little bit more what's happening. And that's what I want to show you now for the last minutes. Okay, and any questions before? Yep. Sorry.
So if I look at NE times SE, so because you have selection coefficient, is that smaller or larger than one? That in terms of effective parameters, is that weak selection or strong?
Yeah, so, okay, that's an interesting point. I was maybe, well, I'm not sure I would have mentioned it later, but I can mention now. So the idea is that when we had yesterday this scaling of NE, right? So the NE was scaled, or you take the N amount of individuals and you scale it by beta square, right? Which increase the population size, the NE. This NE is not scaled the same for selection, and that's what it means. So the NE would be divided only by beta there. So that's kind of the difference, is that it's not only you take the NE of the population and say, oh, I scale my NE by the seed bank and I apply it to the selection, it doesn't work like this because it's not the same scaling.
So NESE is larger.
Yep, it's larger. Yep, exactly. But not as large as if you would be scaled by beta square. Yep. But it is larger than under the right Fisher. Yep.
Times also smaller, probability is larger.
I'll show that in a second. Okay, so that's exactly what we wanted to look, is let's get a bit more insight into what's going on. So we develop our own simulator. That's a little schematic, again, of what we did yesterday, just differently. We have here our samples of the current generation, and they can come from some individuals who were dormant, right? In the past, this is the seed bank, basically. Here we have a fake diploid model. So these are kind of plants which are diploid, but we assume that each of the haploid genomes in them come from different parents, which can come from different individuals in the seed bank. Right? So in a way, we have each chromosome can choose its own dormancy time here. So it's a bit a half model between bacteria and plant in a way. It's a little bit confusing, but we had to do that because we wanted to fit this Chi model where we have this model, but with the diploid model here. Okay, so you can see that you have, for example, this blue comes from this individuals who came from 2 years ago, and this one here, and so on. Right? And then you can have coalescent as we had yesterday. Okay, so these are just a couple of graphs to show you how the model works and that we recover what we were expecting. So here, these are neutral model, neutral simulations. So we can simulate now full chromosomes. So the idea also is because we want to use that to analyze data, right? So that's why we built a full simulator that we can simulate a full chromosome and full genomes so that we can really apply that to our data later, right? So that's the idea. And so what we show here, oh, sorry. So we have here as a y-axis the time to the most recent common ancestor. Again, what do we expect? We expect that if you have no seed bank, this time should be recent. If you have strong dormancy, so here we have 0.25 is this beta, right? B is beta in this case. And so what you see is that you should have a longer and longer time to the most recent common ancestor when beta gets smaller, right? Which is exactly what we had. And it's scaled in beta square. So everybody's happy, right? So we recover exactly what we were expecting from the model. You can show that when you increase recombination rate, the variance gets smaller. That's also expected. I mean, that's also a classic result of coalescent theory. The more recombination, the smaller is the variance because you break the trees. And so each of the tree fits better, let's say, around, well, the variance gets smaller around the mean. Interestingly, you can see that the linkage disequilibrium in a genome, so that means that in a genome, we predict that if you start from a site and you look the linkage disequilibrium away from the site, it's decreasing, of course, in a genome always, but you can see that it's decreasing way faster when you have dormancy, so when B is small compared to the Wright-Fisher classic Kingman coalescent. Okay? So we predict that in species where you have dormancy, and that's what I showed yesterday intuitively from the model, but that's what we see here in the simulation, you see always more recombination and lower linkage disequilibrium than for species which don't do dormancy. And that's because of the scaling of the tree and the scaling of recombination, which are different. So that was kind of nice because we did the model, theoretical model, a while ago, and I think we finally had the means to really show in real that it really works, right? I mean, in real simulations. Okay. Now, for advantageous mutations, we wanted to look first at the probability of fixation, right? So you have two things usually you look, probability of fixation and time to fixation. So how likely is it that an allele will get fixed and how long it will take to do so, okay? So the probability of fixation here of advantageous allele you have from selection coefficient is 0, so neutral alleles to very advantageous alleles. And here we have the mean fixation probability over many repetitions. And you can have here the different seed banks from no dormancy to, let's say, strong dormancy or medium dormancy. And you can see that the fixation probability is increasing. And actually it increases for all possible value of beta the same. So obviously the seed bank is not affecting here the probability that an allele will get fixed. But it doesn't matter. So an allele pops up, it has an advantage. The probability to get fixed is not affected by the seed bank. And you have as many chance to get lost as to get fixed as in the Wright-Fisher model and the classic theory of population genetics. Okay, so the dotted line is the theoretical expectation. I think we use the one, yeah, I can't remember exactly which one, but that's one of the classic expectation that you get. And for higher selection coefficient, you see that it's getting a little bit off, a little bit. Okay, what is more interesting is the time to fixation, because that's what I just talked about before. And so what we do here is we have now the germination rates you can see as the x-axis, okay? And here we have the mean fixation time as a y-axis. So I look how long you wait until the allele gets fixed. It's conditional on fixation, obviously, right? If the allele gets lost, you don't account for it and wait until one gets fixed, and then you count many, many fixations and then look how long it takes until you get fixed. And what we see, this is a different selection coefficient from strong to low. And what you can see is that now when you take the germination rate is very high, so here we have no dormancy, all of those fixation time are very similar. And I think it's pretty clear that the stronger is the dormancy, the more difference there is between the fixation time. Right? So if the seed bank would have no effect, all this line should be straight, right? Basically. So that means the fixation time would be the same as what you have without dormancy for all values of beta or B in this case. And I think it's pretty clear this is not the case, right? And this is here the same but normalized. So we normalize now to have all this value here at 1, and then we normalize all this so that we all have the same for the Wright-Fisher here without dormancy. For all selection coefficients, we normalize the fixation time, and then you can see now there are huge differences. So what does that mean? It means here that when you have dormancy, obviously the fixation time is increasing, right? Compared to what you get into the Wright-Fisher model, which is expecting, because dormancy is delaying everything, because you have this waiting time until everybody who's dormant can get up and resuscitate again. But obviously, this effect is different depending on the selection coefficient. So here we have strong selection, that's the lower line, and you have here really weak selection. So clearly, selection with high selection coefficient is basically still fixing much faster compared to really weak selection coefficient, even though you would not expect so much difference under the Wright-Fisher model. So there is an effect here of the seed bank affecting the strength of selection. And that's exactly mirroring the graph I showed you before from the mathematics or from the diffusion model, where we assume that now we have the scaling of beta. But obviously the scaling of beta, if you remember this formula, was very complicated, right? You had beta, sigma, there were a lot of things in there. And that's exactly what we see here, is that actually the beta is affecting selection, but in a non-trivial way, because it depends on B and or beta and S. And so that's why it's not only an NES scaling, it's obviously a bit more complicated because the NE is scaled by B in a non-trivial way as a function of S any longer. And I don't understand this result still to that day. So the intuition is again that the selection happens always above ground. That's why you have this factor B or beta, but obviously it's a non-linear way in addition. So if you think you have some intuition, I'm happy to discuss, basically. Yep. The one inside, you mean? This or? Yeah, so this is just, this is a view of this year to grow, to make this bigger so that you can see how it looks like, right? Yeah, so the red line is, yeah, so that I think that's fitting to this one here, which is the, no, oops. Yeah, okay, no, so the red lines, they are something else in the paper, sorry, that we calculated later. So just forget about it. There were some other statistics we looked afterwards. Okay, and so maybe I'll just summarize here. The main point is that what is interesting with the seed bank is that they will delay things, right? So what we were expecting naively is that if they delay selection, you have more chance that the allele gets lost, right? Because you wait longer. But actually it doesn't happen because you have the seed bank, so the allele can get protected in the seed bank nonetheless, right? So it has to invade the seed bank to get fixed, but it's also protected by the seed bank against getting lost, because you can still stay in the seed bank a little bit longer and then germinate again, right? So you have these two effects of the seed bank, which are delaying things, but also protecting polymorphism against extinction. And so I think these are two effects which play a role here, basically, on top of the selection. Right? The selective phase itself then is very fast, because then once the allele reaches a critical frequency, it gets relatively fast to fixation. Right? And this, we see that this looks like a normal classic selective sweep that you have seen last week. So this first critical phase is important to understand what's going on if the allele will get fixed or lost. Okay. And so I just want to show a couple of extension of what we can do when you have a cool simulator, because that's the fun part of it. When you do genome analysis, you can now simulate genomes and say, okay, let's say that we have in the middle of the chromosome, so this is your chromosome, right? You see it's a 1 million base pair. And in the middle, I will put one mutation which has an advantage, right? So I will put one mutation which gives an advantage, and I will wait until it goes to fixation in a population. And then I can simulate exactly what is the genetic diversity that is expected around the site on the selection. And this is what is called a selective sweep. And I think you've done that last week, right? Okay, so that's a simulation of a selective sweep. The black one is the one without seed bank. That's the one you have seen last week. And you can see it looks a little bit mashed, right? Why does it look a bit mashed? Because the diversity is not very big here. Right? So you can see there's the diversity, genetic diversity pi here. But I hope it's pretty clear that when you look at the blue line, this is what happens when you have dormancy. So dormancy increases the diversity in the genome. So the whole genome has more diversity anyway, right? That we talked about already. But you can see now that what is really cool is that you have the same signal of selective sweep. But of course, as now you go down from very high diversity to very low, this is a very nice signature compared to that one. Right? So it looks more obvious, so to say. Here you can normalize, if you normalize by the genetic diversity away from the sweep, you're back again and you see that there is a slight difference, but not too much between the two signatures. Right? But in the real life, that's what you see. That's the real data. I mean, real simulated data. Okay, and an additional interesting feature is here now we look at the selective sweep, and we look at the time after the sweep, how long after we can still see the signature. So this is the time 0 when the sweep just happened. Right? So you go to the population, you sample individuals, you sequence exactly at that moment. That's what you see. Right? But then if you come 1,000 generations later, that's what you see. And then it becomes that 2,000 or 4,000 generations later, there is no more signature. Right? That's very known because you have mutations which come and they erase the signature of the sweep. Right? So the genome recovers, and then this mutation has been fixed, and all the mutations around, they pop up, and then they remove the signature of the sweep. Right? So you have a very tiny window under the classic model at which you can detect, during which you can detect sweeps. Now, if you do the same with the seed bank, I hope it's pretty clear that actually what is really nice is that the seed bank is also preserving this signature for much longer. You can see here to 4,000 generations, you still see a big valley of polymorphism and the signature of the sweep. So why is this? Because the seed bank here is again preserving many of the diversity and the alleles in the dormancy stage, so that it takes longer for new mutations to pop up and erase this signature. Right? And so that's kind of, well, I think that's one of the main results that I find is really exciting here, is because now that means that when we look in a genome for selection, if you use, well, you have a species which does dormancy, you would expect to see much more easily signatures of selection and for even longer after selection happens. Right? And I think, well, I don't show this data, But believe me, or I can send you the paper, we did that on our tomato species, which do seed bank. And you can see really nice signature of selection. You can even narrow down the genes really well for a very long time after selection has happened. And I really never believed that when we did the analysis first on the real genomic data. I thought this cannot be true. And then when we did the simulation afterwards, we realized, yeah, obviously it makes sense, right? So that's kind of a nice effect of the seed bank here, that it will help us to find selection in the genome, which I think is quite exciting if you work on many species like plant, fungi, or nematodes and so on. Okay, and I will just keep those very quick because I think it's enough. That's just the proof. Basically, here we use some tools that are used to detect the sweeps, right? So basically, we not only did that, but we thought, okay, let's simulate data and apply the tools that people use to detect the sweeps to be sure that it's really true. And that's what you can show. You can show that with seed bank, you have much more power to detect. And not only you have more power, but you have also you have lower false discovery rate, which is the main point. So that means you're less likely to confound your selection signature with other signatures in the genome. And that's really what is the key point in this method, right? You want to have low false discovery rate and recover your sweeps. So this is one of the methods. It's called Omega Plus, and the other one is called SWED. And these are just two different methods to detect. And we can show that you have way more power to detect selection on the seed bank. One important point here is we have recombination, right, along the chromosome, which means here we assume some kind of classic species like plant or I don't know, nematodes and so on, which do recombination. If you do bacteria, it's a little bit different, right? Because bacteria don't do as such really recombination, so you would have to adjust a little bit what's going on. Okay, and I finish with a couple of wrap-up messages about the seed bank. So I hope that I convince you that the seed bank affects the time to fixation of the allele under positive selection. So selection is slower, but it has a non-trivial effect based on the selection coefficient. The time is lengthened, but is disproportionately affected by the seed bank depending on the selection coefficient. And under the seed bank model we use, which is the weak seed bank, we find that we have high detectability of the genes on the positive selection. We did not test that for the strong seed bank model, right, of bloods. So this we have not done yet. But I would expect it probably also work. What is a bit more difficult now for the strong seed bank model and this island model we discussed about is, of course, that there will be a bit of a mix-up signal between selection and the seed bank, because we have seen the seed bank is generating also this U-shaped SFS. So there is a lot of questions here about how to use this genome data if you're in the strong seed bank model. I would also highlight, especially with bacteria, where you have no recombination, because there will be a lot of effects there mixing up and interacting with each other, which are probably very difficult to disentangle. But I think it's also potentially very exciting with all the genome data we get now to try to work into this. And I will stop there. Thank you. Okay, are there some questions? Remark? I work on plants. I don't want to talk about it.
So what do you think? I mean, in bacteria, firstly, this assumption of having a constant size of the active population seems kind of a very strong one. And then the other thing is that in that moment, like given that they might then reach super large population sizes, also I suppose you assume an infinite size model, and in a way, right? So how do those two assumptions like, do they break your
Yeah. So I mean, I think basically there are two answers. So one is from the mathematics. I think nobody wants to do that, and I think in these papers they usually, well, the size is constant for sure, and they have some integrated, I think, in one of the papers, some finite site, a little bit to look for that, but not in much details. I think the finite site I'm not too worried about, because now there are many models and you can integrate it relatively well, right? And you know how it behaves. I mean, we know how it behaves. We also do it for plants already, because we have seen in the data that it happens, that when you have seed bank, because you have way more diversity, you often actually find this infinite site model is violated. So we have already integrated it. I'll show that tomorrow. I mean, I will not show it, but that's one of the assumptions we use in other models. I think the change in population size is probably more critical. And I guess there one has to do a bit of simulations, I think, to test what is the influence. What we have shown is that the demography is dampened by the seed bank, of course. So if you have change in population size, they get, of course, dampened off by the dormancy, right? Which kind of makes that the population size seems to be more constant, but you could still have large effects, of course. And I think mathematically, at least, this has not been done in these models, but by simulation, I don't think there is much problem to do it. I should mention that in at least the few things we've done with demography is that we always assume that the change in population size is faster than this window M, for example, of dormancy. So that means that it's faster than the length of the seed bank, because otherwise you get all sorts of problems that you have the population size is changing, but your seed bank is not at equilibrium with the population size. And mathematically, that's just too annoying. So that's why we do the separation of time scale and of the demography kind of scale. But I think by simulation, nothing would prevent to do anything, right? Okay, thank you.